Dr. Stephen Streatfield is the Director of R&D and Scientific Affairs at the Fraunhofer USA Center for Molecular Biotechnology. He's going to speak to us today about the traditional approach they have taken to develop a TBV and get it into a clinical trial.
Okay, thank you, Mary Beth. Thank you for the invitation to speak here today. So I'm going to talk about just the early stage development of a vaccine candidate using a novel plant-based platform, and just some of the issues that we ran into as a fairly small organization, just developing a subunit vaccine candidate for early stage clinical testing. So I'll give a little bit of an overview of plant-based transient expression systems, a bit on the selection and engineering of a transmission blocking vaccine candidate. I'll talk about the production and purification of that candidate, and testing it in some preclinical studies, and then producing it at large scale in a GMP pilot plant to generate material to feed into a phase 1 clinical trial. So there's, over the last 10, 15 years or so, there's been a lot of interest in developing new expression systems, recombinant expression systems to deal with some of the issues that have arisen with some of the traditional systems around safety, capacity, response time, cost, and complexity. Plants are one of the systems that have received quite a lot of attention in developing a new production system. Plant-based technologies, they can be broadly divided into stable expression systems, where the nuclear inheritance of the recombinant plant is modified, and then transient expression systems, which rely on binary and viral vectors to transient express targets for a matter of a few days in mature plants. This technology has undergone development away from sort of field-based and greenhouse-based production towards production of plants in contained facilities, and indeed there's quite a lot of work on plant cell culture systems. So, as I say, the switch has gone from about 15, 20 years ago from oral delivery of processed plant material derived from field or greenhouse-grown plant biomass towards a more traditional subunit-based vaccine approach of parental delivery of protein products that are purified from plant biomass grown in contained facilities. This is a much easier regulatory path. It's much more compatible with GMP manufacturing of recombinant products. We have a pilot plant at our facility in Delaware, and there are a number of larger scale production facilities now that have been built in the US and one in Europe. This makes a more straightforward regulatory path. Several different INDs have now been submitted for plant-based production systems, and these have entered phase 1, phase 2 clinical trials. And indeed, with a cell culture-based system, Protalix, within the last 2 years, launched their first product, which was from a carrot cell culture system. So the system we use at Fraunhofer is a transient expression system, uses binary and plant virus-based vectors that are introduced into hydroponically grown plants by a process known as vacuum infiltration, making use of natural pathogens of plants, agrobacteria. So the three main components of this system are the infiltration of the aerial plant biomass with the agrobacteria to get vectors into intercellular spaces in the plant cells, and then the vectors that are generally viral-based vectors that can stimulate very high levels of expression and can move cell to cell within the leaves to generate just very high yields. And then a hydroponic system for the fully contained soil-free production of these plants that allows for the uniform generation of biomass. That's just a video of the infiltration system. Basically, the plant material is immersed in a tank of diluted agrobacterium. This is just a small manual scale. We also have at a pilot plant or GMP production scale. This is all done automatically by robotic systems. So they're immersed in the tank there. I'll skip through this in the interest of time, but this is placed in a vacuum chamber, a vacuum cooled, all the air removed from the intercellular spaces of the leaves, and then when that vacuum is released, the plants suck up the agrobacterial solution, and then the agrobacteria will, through their own machinery, will drive the viral vectors into the plant cells. So just skip over that. This is just an outline of the type of vectors we use. I say they're viral-based vectors, relying on the machinery of the plant virus to generate very high numbers of message, and then to move this message through their movement protein function from cell to cell within the leaves. And then we grow the plants, as I said, in a hydroponic system. This is just an example of a tray of the plants, and Nicotiana benthamiana, which is our host species. It's a relative of tobacco. A very small volume of seeds. Actually, can generate thousands of plants, so it's very little seed storage space required. And we grow them in fully cane conditions with regulated lighting, nutrients, day length, and so on, temperature, humidity. Purification then becomes fairly straightforward. It's a combination of early stage steps that are taken from the food processing industry, harvesting, finely chopping biomass, homogenizing, all using modified machinery that's taken from large-scale plant food processing materials, and then moving into more standard methods for protein purification, clarification through continuous flow centrifugation, depth filtration, typically column chromatography, and then ending up with buffer exchange and fill of the bulk drug substance. So moving on to malaria vaccine strategies, of course this session is about transmission blocking vaccines, and that's the program that we focused on as a vaccine candidate. Number of different candidate molecules, of course for that, PFS 48, 45, 230, 25 and 28 are probably the primary ones. We've done some work with all of these, but our focus today on the program we've taken the furthest, which is with PFS 25. So a number of different approaches we took to, or can be taken in any recombinant system, and the plant system's very amenable to this, to different subunit approaches to the vaccine candidate. One is just to express the native open reading frame in the recombinant system. You also make fusions to other proteins that can stabilize the expression and get higher levels of production, and we've worked quite a bit with a thermostable enzyme from Clostridium. And then the VLPs in the lower left corner, which is again a subunit fusion between the coat protein of the alfalfa mosaic virus fused to the PFS25, and then there's also envelope VLP options where a protein can be expressed with this transmembrane domain and then get budding from plant cell membranes to make an envelope VLP. And most of the work I'm going to talk about today is on the lower left segment, which we found to be the best strategy for PFS25 in our expression system. This just shows the nucleic acid sequence of the target, fairly challenging molecule due to all the cysteine residues. We were expressing it cytoplasmically, so there was no issue with glycosylation. This just shows three different constructs from three of those strategies above: a standalone molecule, a fusion to a carrier protein, and a fusion to the alfalfa mosaic virus coat protein to potentially make a VLP. And that just outlines the VLP structure. You've got the coat protein, and then ideally displayed on those coat surface of each molecule, you'd have the PFS25 candidate. What we actually find is that we don't get full display, we get partial display. I'll go through that in a minute. So we made this molecule in a plant system, purified it along the lines of the general schematic that I showed above, and then we had relatively high, certainly well over 90% pure material. The Kamasi gel on the top left side shows both a fusion and free CP monomer, and a Western blot with anti-CP shows those two bands lighting up. So we've got some free CP, and we also have CP fusion. So what we actually have is VLPs, where we have a proportion of the surface is covered in the PFS25. The two lower images just indicate that we're generating VLPs, both through electron microscopy and through dynamic light scattering to look at the size of the particles. We get pretty uniform sized particles. So we took this candidate into some early stage preclinical testing. This was a head-to-head comparison with what was at that time our lead molecule, which was a fusion to the licanase. One of the alternative strategies that I outlined above, comparing it to the VLP. We'd already shown that the standalone molecule was relatively ineffective in our hands in the plant-based production system. So looked here at a couple of different doses, 1 microgram, 0.1 micrograms, two administrations in a mouse model. And this just shows, this was just showing anti-PFS25 IgG levels. It was really just a target selection exercise for us, and demonstrated that the VLP candidate outperformed the subunit fusion to licanase. Standard membrane feeding assay, I won't go through with this audience, but this was the assay that we used to get a better idea of the effectiveness of the candidates. And essentially in this small mouse study, again, the VLP candidate show clearly greater transmission blocking activity than the subunit fusion. So that was the molecule that we went forward with. We wanted to determine whether it was necessary to have an adjuvant with this molecule, so we did a study with the VLP candidate, both with and without our hydrogel. And the results clearly demonstrated that we did indeed need an adjuvant. We're not an adjuvant production organization, so we just focused on a standard available antigen hydrogel, and went forward with that. And we maintained levels of IgG out to 6 months, and that also correlated well with the membrane feed data, again indicating that hydrogel was necessary with this vaccine candidate, and that we had blocking going out to 6 months. So having demonstrated that early stage data, we wanted to move that into phase 1 and potentially later stage clinical trials. So this wasn't a unique program in our organization wanting to do this. Several of our vaccine candidates we wanted to move forward into clinical trials, and what we were really running into, as many small organizations do, is how do you make material of sufficient quality with your novel production platform. So we were fortunate enough to have a large DARPA funded program at that time, also working with other partners, and we managed to secure the funding to generate or to have constructed a pilot plant, which was the first plant of its kind for the production of plant-made biopharmaceuticals. And briefly, this just shows an area in the top left where we grow the plants. Next along, we have infiltration. So we grow the plants here. We have sorry, when I go back. Okay, in the middle, we have an agrobacterium fermentation room. Bring those together at the top middle for infiltration, and the plants express the protein in the top right. And then the rest of the facility is really protein purification, extraction and purification, much more standard to other systems. So it had a multifunctional capability. We could generate master and work in cell banks of the recombinant agrobacterium, grow the host plants, which are essentially we think of as the reaction vessels for the production of the protein, purify the protein product through fairly standard approaches, and then fill the bulk drug substance. We also needed to set in place a wide range of quality systems to meet cGMP production capability and everything that we would need to feed into the IND applications. Then we ran our plant material through several reproduction runs through this plant facility, showed reproducibility through multiple runs, generating the VLP candidates, just comparing to the reference standard that was generated in our R&D labs. This again just demonstrating reproducibility. And now we're moving that material into clinical testing. A toxicology study was successfully completed, and that material has now moved into safety, reactogenicity, and tolerability testing in a phase 1 clinical trial. Primary endpoint is safety, secondary endpoint immunogenicity. The design of the trial was 4 different doses of the candidate molecule, formulated with our hydrogel. And that study is now wrapping up. So just in summary, we developed a plant-based technology system, both from the point of view of the expression system and also making fusions to a coat protein of the alfalfa mosaic virus. We engineered and expressed the plant-produced vaccine candidate, saw that we got uniform VLPs in this production system, demonstrated in preclinical studies that we had transmission blocking, that our hydrogel was necessary, or an adjuvant at least was necessary, and maintained levels of IgG and transmission blocking activity through 6 months. We then moved that into a pilot scale production facility that we built and using a validated plant, and then the quality systems that we set up, and optimize that process for production of the candidate at a 50 kg biomass scale. Demonstrated safety in a tox study and entered that into a phase 1 clinical trial. Ongoing and future work, well, the clinical trial is nearing its end now, but we also we're looking at further engineering approaches, both to improve the expression and process development to improve the yield. We're also looking at improving the display of the PFS25 molecule on the surface of the VLP, because at the moment, for every 10 micrograms of protein that we're given, we're only given 1 microgram of the 25. So we've looked at some cleavage sites there, taking those out, and appear to get better levels of display with later versions of the molecule. We're also testing formulations moving towards more potent adjuvants than our hydrogel. And then some further assay development work as well. And finally, just like to thank all our partners who have been involved in the studies I showed, conducting a number of the assays, and also funding from Bill and Melinda Gates Foundation through MVI. Okay, thank you very much.
As a reminder, we would like to keep our questions to the end of the presentations, but are there any quick ones just for clarification for Steve?
Yeah, I'm Neville from the Vaccine Research Center at the NIH. So we know that for protection for the pre-erythrocytic stage, you need T-cell responses in the liver. So do you see any T-cell responses?
We honestly haven't looked at this stage. We were just looking for immunogenicity. I mean, the primary mechanism of this candidate, as I understand it, is through generating prolonged antibody responses, and we haven't looked at all at T-cell responses.
Thank you very much.